Hey guys, in this video we're looking at cell biology, focusing on a microscopy practical. So you need to prepare your slide is the first step. If you want to look at a specimen, for example plant or animal cells under a light microscope, you need to put it on a microscope slide first. And a slide is a strip of clear glass or plastic onto which the specimen is mounted, as you can see in the picture here. Let's have a look at how to prepare a slide to view onion cells. So, first step is you add a drop of water to the middle of a clean slide. You cut up an onion and separate it out into layers. Use tweezers to peel off some epidermal tissue from the bottom of one of the layers. Using the tweezers, place the epidermal tissue into the water on the slide. Add a drop of iodine solution. Iodine solution is a stain, and stains are used to highlight objects in a cell by adding colour to them. And then you place a cover slip, which is a square of thin, transparent plastic or glass, to the top. To do this, stand the cover slip upright on the slide next to the water droplets. Then, carefully tilt and lower it so it covers the specimen. Try not to get any air bubbles under there, as they'll obstruct your view of the specimen. To look at your prepared slides, you need to know how to use a light microscope, as we can see here. So you clip the slides you've prepared onto the stage. Select the lowest powered objective lens, which is the one that produces the lowest magnification. You use the coarse adjustment knob to move the stage up to just below the objective lens. You then look down the eyepiece, so use the coarse adjustment knob to move the stage downwards until the image is roughly in focus. Adjust the focus with the fine adjustment knob until you get a clear image of what's on the slide. If you need to see the slide with greater magnification, swap to a higher powered objective lens and refocus. You then draw your observations neatly with a pencil, as we can see here. So draw what you see under the microscope using a pencil with a sharp point. Make sure your drawing takes up at least half the space available and that it is drawn with clear, unbroken lines. Your drawing should not include any colouring or shading. If you are drawing cells or subcellular structures, should be drawn in proportion. Remember to include the title of what you are observing and write down the magnification that it was observed under. Label the important features of your drawing, for example, nucleus, chloroplasts, using straight, uncrossed lines. Quick fire questions of cell and microscopy then, so try these to check your knowledge. You name 5 subcellular structures that both animal and plant cells have. What 3 things do plant cells have that animal cells don't? Where is the genetic material found in both animal cells and bacterial cells? What type of organisms are bacteria, prokaryotes or eukaryotes? And which gives higher resolution, a light microscope or an electron microscope?